It's Biology with Mr. B. Biology with Mr. B. That's me! Hello, welcome to a GCSE revision video about the topic communicable disease. This is a big topic, guys. I think the biggest topic out of all the ones that we do here in paper 1. So I've written down in red sort of the key ideas within this topic, and I'll try to get through as much as I can in my sort of 20-ish minute window. So what a pathogen is, the different type of examples that we can get of pathogens, the human body's primary defences, what stops those pathogens getting into our bloodstream, and then if those pathogens get into our bloodstream, how our phagocytes and lymphocytes and memory cells hopefully will go about combating and destroying any of those pathogens. We can link that idea of memory cells to vaccination, which is, if you're watching this in early 2021, very relevant to the current news situations. And we can link that again to drug testing, how you go about testing any new sort of product, any new sort of drug, to ensure that it is safe and effective. There is also a required practical in this topic, where you grow your own microbes and you might test at different antimicrobial properties to see which ones may kill the most microbes, and why and how. So here goes. Let's start straight away with the key definition of this topic, the definition of a pathogen. It upsets me greatly, deeply, on adverts whenever they talk about kills 99.9% of germs. No, germs, no, that is not a scientific term. Don't use it. You mean to say pathogen, because a pathogen is a microorganism that causes disease. I mean, in your body there are more microorganisms than there are your human cells, and 99.999% of them are your natural flora that you actually rely on to survive. But every now and again you get a microbe that causes disease. That's the one that your body will want to kill. That's the one you don't want inside living things. And that's the one that we need to deal with. So there are 4 different types of microbes that could cause disease. 1, 2, 3, and 4. The first are bacteria. The second are viruses. The third are fungi. Great at a party. Fungi. Sorry. And the final one are protists. So within the spec points for this topic, they give you a couple of different examples for each of these. Well, there's only one example for the protist, that's malaria. That is in the spec. You will be expected to talk very briefly about, for example, what salmonella and gonorrhea are, what malaria and tobacco mosaic virus are, what rose black spot fungus is. But in the nicest possible way, the spec points are so beautifully clear with exactly what you need to know, so I'm not going to go into that at this point. I'm going to save myself a bit of time there. What I am going to talk about, though, is specifically about bacteria and viruses. You need to know generally how these make you ill, how bacteria go about actually making you feel poorly. And for bacteria, it's quite straightforward. They make and release toxins. Bacteria live out in the open, so they live usually with where you have tubes, hence why respiratory infections, digestive infections are the most common. Some bacteria, pathogenic bacteria, make toxins. That's a posh word for poisons, and those toxins can damage nearby tissue and nearby cells. Viruses don't live out in the open. Viruses live inside your cells. So they actually enter your cells, your human cells. And they actually go inside your nucleus and your genetic machinery, and they kind of take it over and they use your genetic machinery to reproduce. And they reproduce again and again and again and again and again until that cell is so full of viruses that the virus literally bursts out which of course damages the cell in the process, and then they go and infect and enter neighbouring adjacent cells. So bacteria live out in the open, release toxins. Viruses live inside your cells, reproduce rapidly, and burst out of the cell in order to infect others. And by bursting out the cell, clearly it's going to damage it. So that leads me to one final point about this, and that's our drugs called antibiotics. Antibiotics are a drug that your doctor can give you to help kill pathogens. And they will give you antibiotics to kill bacteria. Because bacteria live out in the open, so if you do take some antibiotics, it's easy for the antibiotics to find the bacteria, and by attaching to the bacteria, the antibiotics are able to kill them. Antibiotics cannot work on viruses. Because viruses are inside your cells, the only way to kill them would be to kill your own cells. Now, the only treatment that regularly does that on purpose is, for example, things like chemotherapy and radiotherapy, and the symptoms of doing that are extreme. You would only kill your own cells purposefully if there's a real emergency situation. So antibiotics cannot be used in viruses because viruses live inside your own cells, and antibiotics don't enter your cells and they don't destroy your own cells. That would be counterproductive. You don't need to appreciate how fungi and protists make you well. So yay, a bit less to learn, but there you go. Right, so next job then. So if we've got these bacteria that can make you poorly by releasing toxins, viruses that live inside your cells burst out, damaging cells, it all sounds really, really nasty. But the good news is, your body has its own natural mechanisms to, number one, prevent pathogens entering the body, and number two, kill them quickly if they do. So I'm going to draw my mate, Bob. That's Bob, he's my mate. Right, so and what I'm going to do on Bob is label as many of those primary defences as I can. Okay? So I want as many primary defences as possible, which are going to be mark worthy as well. The first of which, I'm going to label as eyes. I'm going to write tears. Tears actually have antiseptic properties. They contain enzymes that can digest pathogens. And that hopefully will prevent pathogens entering your eyeballs and getting into your blood and your body that way. Next I'm going to label is skin. I'm going to do that at his leg to give me a little bit of room. So skin. And with skin, it is a barrier. That's the key word. A barrier to block, to stop pathogens entering. And even if you break that barrier with damage, let's say you get a cut, well, that's when you get scab formation, which is because of platelets, which is that component of the blood, aren't they, that do blood clotting. So there's a nice link to the circulatory and health topic here as well, isn't there? The idea of platelets taking part in this protective mechanism. The final ideas are with your respiratory system and your digestive. Because I'm sure you can appreciate, every time you breathe in, well, you're very likely to be breathing in pathogens. Every time you eat food, it's very likely there might be some small contamination. And you need to deal with that. Your body needs to deal with that to ensure those pathogens don't get into the blood. So I'm going to do it as a kind of a story, starting with breathing in harmful pathogens. So here's what happens. If you breathe in harmful pathogens, so let's do an arrow to where the respiratory system may be, top half of the stick man. Sticky mucus will trap pathogens, dirt, dust in your trachea and the bronchi. Now, yay, it's trapped. Problem is, mucus is actually a fantastic environment for bacterial growth. So if it stays in the mucus, all it's going to do is the bacteria is going to reproduce really quickly by binary fission. That's how they reproduce. And you're just going to get a massive infection. So yes, it's trapped. So the next job, you need to get rid of the mucus. Yeah, we need to push that mucus somewhere else. And that's where we have cilia. Cilia are the cells lining the trachea and the bronchi that kind of have little arms. Okay, they're not arms, are they? But they look like little arms. And these little arms waft. And in this case, they will waft upwards to the back of your throat. So the cilia waft the mucus with the trapped pathogens towards the back of the throat. And they will do that constantly. They are constantly using energy from respiration to waft that mucus upwards. And when it gets to the back of your throat, you will naturally swallow. And hydrochloric acid in the stomach will denature all the proteins that arrive there. And because living cells and pathogens are primarily made of protein, that will destroy the pathogen. So this lovely story linking together the mucus, the cilia and the stomach acid, that is a lovely, lovely linked idea that's going to get lots of marks. The skin is obviously a nice idea as well. Just make sure you use your keywords. If you can link it to platelets as well, that's a nice link, a good idea. And tears are usually just that little extra point you can put in if you need another mark here or there. Right. The next little journey within this conversation is, all right, even though you've got these amazing primary defences as an animal, as a human, there's always some pathogens that get through. There's always some that get into your bloodstream. So what does your immune system now do about it? So your immune system in your body are your white blood cells. And white blood cells do three main things. Some white blood cells engulf and digest the pathogens. I mean, they literally go up to the pathogen and go nom 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 nom. So engulf it, that means take it in, and digest it. They'll use enzymes to digest. Those types that engulf and digest are phagocytes. Nom nom nom. So they're not specific, they will engulf and digest anything, and not just pathogens for that matter. If they can get told, they will get told to engulf and digest old cells, dead material, other things within your body, but they will also do it to pathogens that shouldn't be there. The second type of white blood cell produces antibodies. Not antibiotics, no, this isn't a drug. An antibody is a very specifically shaped protein. And antibodies can actually kill pathogens. And I will talk you through, because that's where we're moving on to, I'll talk you through how they do that just after. But white blood cells that produce antibodies are called lymphocytes. The third thing a white blood cell can do is produce antitoxins. So, which is obviously absolutely fab if they're trying to combat bacteria. Because if bacteria make toxins, then these antitoxins will neutralise the toxins produced by harmful bacteria. So they don't actually kill the bacteria, the antitoxins, but they do neutralise the toxin. So they'll reduce the symptoms that you have. So what I want to focus on is how this happens. Because how this happens, what the lymphocytes does, that will result in long-term immunity because of the production of memory cells. And it's what vaccinations rely on. So lymphocytes. So let's imagine we have a pathogen. This is a bacteria. I'll label it bacteria because my drawings are that bad. I do need labels. And on my bacteria, I'm going to have little triangle protein markers on the surface. And these triangle protein markers, I'll colour in red, they are called antigens. And they are on the surface of the pathogen. The white blood cell, the lymphocyte, I'm going to use a lymphocyte that has a very special lobed nucleus. Learn more about that one at A level. The lymphocyte, as we said before, will make antibodies, which are just little proteins. So the antigens are little proteins on the surface of the cell membrane of the bacteria, and the lymphocyte makes little proteins called antibodies. And the lymphocyte will actually make lots of different types. I'm trying to draw different shapes. That's done quite nicely then. Now clearly, looking at the shape of the antigen, not all of these are going to bind, are they? That one won't, that one won't, that one definitely won't. The one at the top won't, but this one will, won't it? That one will have a correct shape. And the ones that the lymphocyte produce that have a specific shape, complementary to the shape of the antigen, will be able to bind. Like this. And when that happens, it will kill the bacteria. How it does that, we don't need to be aware of. That is A level, but it does involve effectively bringing over phagocytes, bringing over the ones that engulf and digest. By having the antibodies, it makes it very sticky. All these bacteria with the antibody will stick together, form a clump, and in white blood, the phagocyte will engulf and digest the big clump. But that's A level. All you need to appreciate is when the specific antibody binds to the antigen, that's it, the bacteria is going to die. So the lymphocyte, though, isn't finished. It will remember the correct antibody, the correct specific antibody. And therefore the lymphocyte will become a memory cell where it will stay inside your blood for a long time, forever, and it will be able to rapidly produce the correct antibody, the correct specific antibody, if you get reinfected. The first time you get like a pathogen in your blood that you've never seen before, it takes about a week or so for the lymphocytes to make the correct shaped antibody that will kill it. But if it enters again a year later and you've got your memory cells, because it already knows the antibody to make, it makes them instantly, and you would actually kill the bacteria, kill the pathogen, usually before you even notice any symptoms. Most of us will have had chickenpox. Took us probably a good week or so to get better when we were little. And that was your lymphocytes finding the correct antibodies. Probably every couple of years or so, the chickenpox virus probably does end up inside your blood. But I'm going to presume very, very few of you have had chickenpox twice because of your memory cells. Because the correct antibody was created so quickly that the virus hasn't even made you ill. You've not even got symptoms. Now, vaccinations work exactly the same way. What a vaccine does, I mean, if I think of the measles virus vaccine as an example, what they literally do for the measles virus This is my measles virus. And it will have antigens on its surface. I've just made that rubbish. Hold on. Measles. So they actually inject you with an actual measles virus. But what they've done to the measles virus is they've attenuated it. In other words, they've removed part of its genome so that it can't reproduce. And therefore it can't make you permanently poorly. It's why when you do get the measles virus, you will potentially get, in about a week or so, you might start to get the rash, which is your immune system fighting it off. But you don't actually get ill because you have a measles virus that doesn't actually make you poorly. But it does have the correct antigens, which means you do get lymphocytes trying to create specific antibody, and when you do make the correct antibody, they'll become memory cells and rapidly produce the correct antibody if reinfected. All a vaccine is, all they inject you in with is a weakened or even a dead version of the pathogen. They could even just inject you with the antigens, and that would still trigger lymphocytes to make the correct antibody. Right, last little bit then. If we're developing vaccines, if we're developing like new drugs, like antibiotics to try and treat diseases, it needs to be highly regulated to ensure that it is safe and effective. So there's a lot we could talk about drug testing, but I'm just going to focus on the 4 key stages of drug testing. The first is preclinical trials. Preclinical means before using humans. So a preclinical trial will be using cells and maybe using animals, flies, mice, etc. So before we are testing on humans, what we're actually looking for is efficacy. Does it work? Does your drug destroy the cell, or do what it's supposed to do at a cellular level? And we're also looking to see if it's toxic or not. Is the drug toxic? Does it kill cells that it doesn't need to? And if it passes those stages, it will begin to be used on clinical trials. And both step 2 and 3 are clinical trials because they use humans. For stage 2 though, they will actually only use a small number of healthy volunteers. And they do that because they're trying to look for side effects. You don't want to use an ill person to look for side effects because you might only see their illness. Likewise, if they're already on medication, the medication they currently take may distort the sort of side effects that they may be getting from the new medication. So the healthy volunteers, male as well, because of the risks potentially to any female that may be pregnant. So yeah, looking for side effects. And if it passes that, that's when you'll use a large number of patients, so people with the illness, with the condition, and you're looking to find the optimum dose. How much do I use for different types of people? It's in this stage, stage 3, where you would use a control group which receives a placebo. So a placebo is a fake drug. So it looks like the drug, tastes like the drug, but it doesn't contain the active ingredient. So a control group would be actual people, actual patients who just receive this placebo. But it's very, very important that the patients don't know if they've got the real drug or if they've got a placebo, because that ensures there's no psychological effects. When neither the doctor or the patient knows who's got what, it's called a double-blind trial. The final stage is peer review. And this will be done by a highly independent body, not the government, not the drug company. And what you're doing by having a peer review, you're ensuring that there's no bias. And the drug will only be approved if this independent body agrees, yes, it does work. Another way to remove bias, by the way, is the double-blind trial. If no one knows what they're taking, you can't be biased and think, oh, they've only had a placebo, they definitely won't feel better. Well, actually, they might feel better. Anyway, so thank you very, very, very much for watching this video. I know it was a little bit longer than usual, but it's a big topic. So take care, be good. And regarding the required practical that I've not been able to get onto, again, do have a look at my revision booklet. It's really good. Okay, bye-bye. Take care.